Okay. I think that's recording. Okay, so let's just go over these, see how you did. So briefly explain the differences between endemic, epidemic, and pandemic. Endemic means something that's always kind of, it's there in the populations, but not at unexpected numbers. An epidemic is when there's unexpected rise in numbers in a particular location, and a pandemic is that it's worldwide, that we're seeing numbers increasing around the world, right? So you said something like that. What do the abbreviations CDC and WHO stand for? Centers for Disease Control and the World Health Organization, right?
Yes.
Okay. Normal flora will often be found contaminating. It's not really contaminating, it's supposed to be there, right? But anyway, contaminating a culture. What can you do or use to determine if the microbe is there as part of normal flora or if it's causing disease? You could have answered this question with one word: quantification. That would have been the word you could have used. And I would have assumed that you knew what that meant, that you're quantifying how much normal flora as compared to the potential pathogen and use it. So I did spend some time with that. We all go with that question? Good, great. Name the most common bacterial cause of urinary tract infections in post-pubescent women. Escherichia coli. In the prior question, which was E. coli, please answer why this microorganism is the most common cause. Why is that? Okay, so you gave a specific reason why. Because
I put the areas are close together.
See, that's true too. You know.
Well, I put the so close together.
Okay, so all of those things would be good. I can hardly wait to see what else is written.
I didn't want to be too specific.
Okay, well that's okay. That's okay. But actually, what I was getting to is that it is an opportunistic pathogen. It can be part of the normal flora, and it can take the opportunity, because we know what opportunistic pathogens mean. You're not catching them from somebody else, are you? They're part of your normal flora that take the opportunity to cause disease. And there are lots of things that people do that make them more susceptible. And we'll talk about, certainly the hygiene issue and like wiping, and also urinating frequently, right? And urinating after intercourse, and all of those things can contribute to having this microorganism become an opportunistic pathogen, right? I can hardly wait. What microbe is the most feared in the burn unit? And Brandy, what did you write? Oh, because I said, oh, never mind. I thought I also put in cystic fibrosis patients, but I didn't add that. Which one is the most feared in the burn unit? It's Pseudomonas aeruginosa. And I meant to put also in cystic fibrosis patients. Sorry, I thought I put that in there. I hope you got it right. And why is it the most feared? Because it's really difficult to treat. It's a really resistant microorganism. It's really difficult to treat. And I may just omit that because I don't know if I talked enough about that. But that's why it's feared in the burn units, is because it is so difficult to get rid of. What is meant by a pure culture, we can thank Brandy for. So I hope everybody knew that meant that there's only one microorganism growing. It's in pure culture, growing on BDS. And again, just in case, because I had some people who maybe just were a little bit confused about that whole conundrum between infectious diseases and microbiology lab and diagnostic medicine. The conundrum is that infectious diseases kill you quickly, or can, but in the lab for definitive diagnosis, it takes us some time. Time is of essence, but it's taking some time to get pure cultures and then doing testing to get to that definitive diagnosis. So just as a reminder of that. Anyway, thanks for being here. You got some points for being here. I appreciate it. Anyway, so good. Did we have any discussions about any of those? Did they seem fair from last lecture? Yes, Jarrett?
Could you go over in a little bit of detail quantification?
Okay, so quantification means that you're assessing how much of something is present. So to quantify is to say how much of something there is, right? So if on a culture there's not much normal flora, but there's this abundance of a potential pathogen, then it's an opportunistic pathogen.
So quantification is just looking at
Quantification just means the amount. You're giving like a 1 + 2 + 3. You're rating the amount of it. You're quantifying, right? Can somebody help me with this? Give me another that might help Jared. And sometimes y'all will say things that make sense to other people. And like in my own mind, everything's making perfect sense to me. To us, Siri. So anyway, but y'all say something. So what's another thing for it?
It's on the plate. If it grows,
Well, wait a minute. Everybody has to be listening. So what do you say? What? Hold on. Do you think you get it now?
I know the definition of all that, but I was wondering if there's like a process about
A process.
it to go about it.
I might not understand what you mean by process. We grow microbes in the micro lab. We actually have to grow them out. We don't always have to do that. We'll talk about other things that we do, but the things that we do grow out, we're literally growing them out and quantifying the amount that's present. So we're actually assessing how many of them are present from that original set. So it's just that quantification that's given, the amount that's given. Do you think you get it?
Yeah.
You too? Okay. All right. So,
Okay, so here we go, guys. We didn't finish chapter 2. I thought we were about finished. We talked about the 5 eyes, though, didn't we? And we were actually talking about those 5 eyes. And there are a couple of things I do, even though most of you, I mean, I don't think anybody's going into
diagnostic medicine into the laboratory. I still think there's some things that you need to know that we do. There's different types of media, and you all are going to get to play and use some of this media. And there are just some basic things I want you to know about it. We have what's called enriched. We have what's called, and these are different types of media that these specimens from patients will be placed on, right? So specimens, whether it's sputum, whether it's pus from a wound, whether it's stool cultures, whatever, they're going to be placed onto media to be potentially growing out any pathogens that are present. So some media is just enriched, and that means that it's going to grow a variety. When that means that it would grow, it's just going to have plenty of food in it, and it's going to grow a variety of microbes that might be present, right? So it's called enriched media. I'm going to just tell you what I want you to know about these different types of media. Then we have some media that is called selective. And media can be made selective to only grow one type or a few types of organisms by adding things to them, like antibiotics. Antibiotics that would destroy one whole class of microbes, but would allow another class to grow. So enriched media can be made selective by adding something to it, like antimicrobials that would get rid of some of that normal flora for you so that you could see potential pathogens. Does that make sense? Because we said that a lot of times the specimens that are coming from patients that you all are going to have to collect from patients, you know that they're going to have a ton of normal flora on them, right? Because there's a lot of normal flora everywhere. Not everywhere, but in most places, right? So the lab is going to have to do something to try to get out potential pathogens. So before I go any further, I need to tell you about something else. I just told you all that you can tell when something is an opportunistic pathogen by quantifying it, didn't I? And I told you that opportunistic pathogens or microbes that are always kind of in our environment. They're always, on and off of us, and they're part of us, but they should be in very low numbers. So if we see them in higher numbers, we know that they're opportunistic pathogens. Fair enough? Have I told you all what a true pathogen is? Let me tell you what a true pathogen is. So you all would be able to discuss the difference between an opportunistic pathogen and a true pathogen. A true pathogen is one that should never be present, not in any amount, ever So if it's there, and I don't care if it's hiding, and it's just a little bit there, it's a pathogen. It's a true pathogen. Let me give you an example, one example, okay? By the way, when I give you all examples, make sure y'all know those are just examples. It's not exclusive, you know. But here's an example. Here's one example that I think you'll all kind of get. You know, y'all have all heard of gonorrhea, right? Let's write down that gonorrhea is caused from a bacterium known as Neisseria gonorrhoeae. Neisseria, starts with an N, gonorrhoeae is the species. It's a bacterium. So can we treat it with antibiotics? Sure, we can treat it with antibiotics. This is actually, where have y'all heard of Neisseria gonorrhoeae? What does it cause? It causes gonorrhea, which is actually a what kind of disease? So we think of this as being a sexually transmitted disease, which is true. And the numbers of these diseases, this is actually on the rise right now. We're seeing more of this. And something of concern about Neisseria gonorrhoeae right now in the infectious disease community is that we're seeing stereotypes of this that can't be treated with anything. We've got some that are incredibly resistant to the antibiotics that we have available. So is that of concern?
Yes.
Is the CDC keeping account on that
Yes.
and seeing where those outbreaks are? Yes, they are. Thank goodness for the CDC. Okay, so this is obviously, I'm telling you, it's a reportable disease. So this has to be reported. It's one of those reportable diseases. But let me tell you something. You can have just a little bit of gonorrhea and be okay. If you have any gonorrhea, that's a true pathogen. You have disease. Do you always have symptoms with gonorrhea?
No.
No. And when you're getting the specimens that gonorrhea may grow from, are those specimens usually going to be from areas that have no normal flora? Or are they going to be from areas that have a lot of normal flora? They're going to be from areas that do and should have a lot of normal flora, because the normal flora is protected in these areas. So, okay, so you've just gotten a urethral swab. Can you picture that? That's got all this pus, green, gooey, brown pus. You've got a vaginal swab that has the same color pus. And you expect there to be a lot of normal flora, right? You expect there to be a ton of it. But what if there is just a little bit, even one colony, just enough to make one colony of gonorrhea? Would you want to miss that? Because all the normal flora is growing? No. So what kind of medium are you going to use? An enriched or a selective? Selective, because you're looking for a true pathogen. Same thing with stool cultures. We can all picture what stool is, feces, right? So you got stool. 95% of the dry weight of your stool is bacteria. Are the bacteria in your stool important for you to be healthy? Yes, it's filled with microbes. It's supposed to be. You want there to be all these microbes. But what would happen if maybe you had, and y'all have heard of something like salmonella, haven't you? And salmonella might not be in very high numbers. The quantification might be really low, but it's a what? True pathogen. And do you have to be able to get through the forest to find that little tree, right? You have to be able to. So what kind of media do you think we use on stool cultures? We use selective. We try to get rid of anything, a lot of it. Are any of these medias perfect? No. So what's the most important thing that's happening is the primary care providers, which are nurses, collecting those specimens, making sure they're collecting them correctly in the right whatever containers they need and getting them to the labs on time. And then the lab will take over and do all this selecting process as far as like what media to put it on. But does it make sense that for something, we need enrichment, certainly. You all will be using something that is a basic enriched media. It's called sheep blood agar. And you know why it's called that? Because it has sheep blood in it. It really does have sheep blood in it.
We used to have sheep. Did you know that we used to actually have sheep at EBA? We had this one person, I remember her very well, who made all of our media in-house. Like she actually poured it all and mixed it all. It was really cool. What'd you say?
I said very organic.
It was so cool. But then I remember when, we started purchasing it outside her Gloria's little job. She sort of lost her little job, but she didn't lose her job. I mean, we kept her there, and she just took on some other responsibilities. But I used to love going in and watching her make the media. Anyway, it has sheep blood in it, and it has some other nutrients in it as well. And it will grow a variety of things. Sheep blood agar will. Y'all will be using sheep blood agar when you're culturing it. And so it's going to grow a ton of stuff, right? And you're expecting it to grow most of the normal flora that you have growing. And you'll see different colony types you'll know they're different organisms. Yes.
Why is sheep blood in particular?
Well, that's a very good question. You could use other, but that was just, it's something it's got like vitamin K, and it's something they call hemoglobin. I forget what it is, but it's just a perfect medium. So they do use the sheep, and that's usually what they use. Now, we can make it even more enriched by doing something like heating the sheep blood and those red cells lyse, and then they release these factors into the medium, and that's called chocolate agar. It's called chocolate agar because when you heat blood, it turns what? Instead of red now, it looks what? Brown. So it's called chocolate agar for that reason. But which is the most enriched? The sheep blood agar or the chocolate agar? The chocolate is most enriched. Now, there's some microbes, and I want you to write this down. Some microbes actually require chocolate agar to grow. They have to have the more enriched agar, or they can't grow. I'm going to give you 2 examples of those. These organisms are called fastidious, meaning that they're picky, meaning that they're like little picky princesses. They require that chocolate agar. Here are the 2 examples. One of them I just gave you for something else. It's called Neisseria gonorrhea. Neisseria gonorrhea has to have chocolate agar, it won't grow. And here's another bacterium. It's called Haemophilus, starts with an H. Y'all can Google these, but Haemophilus influenzae. And that's the species, is influenzae. Please don't get that species of that bacterium confused with a virus that we call influenza, right? So Haemophilus influenzae. Have you been introduced to Haemophilus influenzae yet in this class? I know you have in A&P, but have you in this class? No. So in this class, what I'd like to introduce you to this bacterium, Haemophilus influenzae, is that this can cause a variety of diseases. It can cause some of the things more common that are associated with it are middle ear infections. Middle ear infections. Conjunctivitis. What is conjunctivitis? Pink eye. Pink eye. It can cause pneumonia, and it can even cause meningitis. So, we have a sputum culture on a little kid, or anybody actually. We have a sputum culture on a little kid, and we know that, whatever. Do you think we're going to put a chocolate agar plate in with that setup? Do you think we would want to if we know we've got an organism that can cause pneumonia that has to have the chocolate agar? Or you're going to miss the growth? You can't, you would miss it. All right, so like I said, y'all aren't going into diagnostic medicine in the lab, but this is what's going on in the lab. So when they see where you got the specimen from and where it's collected from, they know that there might be some true pathogens hiding, or true pathogens that are fastidious. And so they're going to decide which media to use based on that, based on whether they've got to be selective in trying to get rid of some normal flora so they can see true pathogens, or whether they may see fastidious organisms that are picky and need the more enriched medium, like what agar? Chocolate.
And chocolate is simply sheep blood that's been lysed. Yes.
Would chocolate agar still work to for normal flora as well?
Of course. Yeah. And so what we can do, it will still work for normal flora, but what we can do is actually see that on the sheep blood agar plate and the chocolate, looking at them both, that there's certain things growing on the sheep blood, but there's one other little thing growing on this chocolate that was not on the sheep, and we know that means that it's fastidious. And so we can see the colony is on that one, but not on this one. But we can also add, and what we do for urethral swabs and what we do for vaginal swabs is we add an antibiotic to the chocolate. And that makes it now a not only really enriched media, but also a what? Selective media. So anytime there is a chance that Neisseria gonorrhea is there, we are going to use a selective chocolate agar. It's called Thayer Martin. Y'all don't need to know that, but it's called Thayer Martin agar. Yes.
So the chocolate agar is the sheep blood, and I assume you're comparing it to the specimen that you have.
When they're growing, yeah, you would always, if you had them both there, you're going to be comparing, looking at them and see if there's something on that chocolate that wasn't on the sheep blood. And we also, in a micro lab, you get trained to recognize certain colonies, like what they are. It's sort of like y'all walk in here and I've kind of gotten your names down, right? Because you each look different. To a microbiologist, colonies are the same. You get to know them, and you get to know which one looks like Staph aureus, and which one looks like Strep pyogenes, and which one looks like Neisseria gonorrhea, and which one. So you learn how the colonies present themselves, which we're going to talk a little bit about today. But that's not the only thing you do to identify them. You're going to still go through getting them in pure culture and then doing the biochemical testing that you need to do to identify them, right? Did that answer the question? Forgot the question.
What is a chocolate agar exactly? You said it's sheep blood. Is it
So
always sheep blood?
yeah, so it's heated sheep blood though, because it's heated and now it's more enriched than regular sheep blood. So when we say that there's enriched agars, some are enriched, they grow the most things, but some are even more enriched, like chocolate agar that grow even more. So chocolate agar would be the most enriched, if you would, you know.
Or does it just have to be sheep blood? The other thing.
No, it's sheep blood. That's lice. Yeah. Yeah. Okay. So this is what these different agars are talking about, and how they can be selective. So because we expect many of our specimens to be mixed, we expect them to be, and it's all about getting something in pure culture as quickly as possible. So when we think about a single colony, we're going to know that they are of one particular organism that's growing, right? And so we can see that there's different things right here. We incubate them, but we need to get them out in pure culture. So we're going to use everything we can to try to do that. Now, some of you haven't seen agar plates before, and these are good pictures. These pictures show you. You can, just looking at this picture, can you tell that this one that looks really red? And by the way, some of the way that we can make media, and I didn't tell you about this type of media. Yeah, and I want you to write it down. So we have enriched, we have more enriched, we have selective, right? But we also have media that's called differential media. Differential. And differential means that there's something in the media that can help you to really quickly differentiate between colonies, between microbes. It's differential. So let me give you an example of this one that we're seeing right here, which is kind of nice. I think. Let me see if it's better. What'd you say?
Quickly differentiate what?
Between different types of organisms, so you can see that they look very different. All right, so I'm going to take some liberty with this one. Can everybody see this top plate? Can you see this top plate here? Can you all see that there's a ton of stuff growing right here in that first quadrant that we said, right? And everything's growing on top of each other. But we said we would sterilize the loop and streak it out to here. Then we sterilize it again and streak out to here.
hoping that whatever's predominating won't be competing with so much food, so there'll be nice isolated colonies. Do y'all see the isolated colonies here? Can you see that? You can see that? When you all look at this plate, can you see a distinct difference? One of these colonies has pigment. It's red. Do you see that? One of them is kind of is yellow looking. Do you see that? And then one of them is kind of a gray translucent looking. These colonies are different microbes. They're different microorganisms. Now, to get them in pure culture, we're going to take a sterile loop and go down, just down and up, because you can get millions, and you'll see this when y'all are doing this in lab, just going down and up and just barely touching the top of that colony, you really have that many microbes of this organism. And you can take it to another agar plate, and then after it grows, it will be pure, right? We would take this one and kind of do the same, and this one and do the same, except we really wouldn't in a micro lab, because let me tell you why. They're so differentiated that I would be able to tell right away which ones were just part of normal flora and which ones may be potential pathogens. And would we need to isolate all the normal flora? No, we don't even care about it. We just want to quantify it. We're just going to say on that report that there's 3+. What would I call that 3+? Because it's growing out to here. I would say 3+ normal flora, but then I've got this other thing I need to look at, right? This media can be made differential by just adding a sugar to it. And some of the organisms can actually use that sugar, and when they use the sugar, it's going to change the pH, and they're going to have a color that precipitates out. So we can make media differential just by adding sugars. We can make it differential by doing a few other things as well. And all it means is that you'll be able to easily pick out different colony types because it's differential. Now, what did I tell you sheep blood is? Sheep blood agar. It's what kind of medium?
Enriched.
It's enriched. But sheep blood agar can also be differential. Do you know why?
It's got blood sugar in it. Well,
No. No, it's because regular sheep blood, y'all know it just looks red, right? It just looks red, and you've got colonies growing on it. So you may have a ton of colonies because if you did somebody's throat, they're going to have a ton of colonies growing. But some of the colonies will actually be lysing the blood that's in the agar. They will be lysing the blood. It's called being beta hemolytic, but don't write that down. But you can look at colonies and see that they have this zone where they ate it completely clears the blood out, and it looks clear. So that colony is hemolytic. If we broke down hemolytic, what does that mean? Heme means what? Lytic means what? Lysis. Hemolytic. Have you heard hemolytic before? You should know that word if you haven't. Hemolytic means lysing blood. Do you think that bacteria can just lyse the blood that's in a sheep blood agar plate, or do you think it could lyse your blood too? Do you get what I'm saying? So we can see the colonies that are non-hemolytic, they're not going to be having any little zones around them. Versus colonies that have a hemolysis around them. Some bacteria are what we call alpha hemolytic, which means that they'll do a partial hemolysis. But is that helping us to differentiate them? Is it? Is it, guys? Yes, it is. So if a bacterium has the ability to lyse blood, if it has that ability to lyse blood, do you think that contributes to its virulence? Yes, it does. It definitely contributes to its virulence. So bacterium that y'all already have in your list, and I'm never going to ask you whether something's beta hemolytic or not or whatever, that's for the lab. But maybe it just helps you to understand why Staph aureus is so awful. Because guess what it has the ability to do? Lyse blood. It's beta hemolytic. Guess what Streptococcus pyogenes can do? Actually, most Streptococcus can do this. Lyse blood. So that adds to their virulence factors, making them a little bit more dangerous than some of the other things that are there that can be there as part of normal flora. Yes.
I'm sorry, could you define lysis?
Lysis means to split. If you ever hear lysis, it means to split. So something that's lytic means it's in the process of splitting. Hemolytic means it's splitting blood cells. It's splitting blood cells.
I was about to ask the same question too.
It's a good question. It's a good question. And if I ever say something like hemolysis or something like that, because sometimes I just think that you have it or you know it. Please stop me. I would rather stop me and everybody be on the same page. Escherichia coli. Escherichia coli has the ability to lyse blood. It has the ability to do that too. Some of the serotypes do. So, that's one. I told you all, and y'all are feeling like maybe you're adding a lot to your list, but it's not that many microbes that really are so dangerous, but you do need to battle most times. Yes.
So this is sheep blood differential too, like how?
Because you can see whether one is hemolytic or not. When you look at a colony, you can say that one's beta hemolytic, but that one isn't. So is that differentiating between the colonies? When you all look at it's so hard for me because you're not looking at plates, but when you are looking at your plates, you're going to see a ton of growth. You're going to think, oh, was that really in my throat or up my nose or between my toes? And yes, and you're not going to see just one colony type. You're going to see a lot of different colony types, right? And sometimes the media can really help you out with that, especially if it's differential media. And you will be mostly using, sheep blood, okay? And that's mostly just because I'm limited in the funds I have. All right, so inspection and identification, because we said it's all about getting the identification of what we're dealing with, right? Because then we'll know how to treat. Sometimes when we're treating, we're guessing, and we're trying to treat with broad spectrum drugs that are really not very safe for the patient, but we're scared they're going to die if we don't, right? So what we want to do is, as quickly as we can, narrow down the drug choice so that it won't be as toxic to the patient, and it will be just hitting a perfect chemotherapeutic. In a perfect world, a perfect chemotherapeutic would only hit and hurt the pathogen, but would leave all your normal flora alone and would leave your own human cells alone. Do we have any perfect chemotherapeutics? No, we don't. But what we do when somebody comes in and we think they've got something that might be taking them out of this world, we have to treat with broad spectrum drugs, chemotherapeutics, until we get our definitive diagnosis. Remember, why is it important that when somebody comes in and they look like they're crashing on you? I mean, they really look like they're crashing. They may have a high fever, their kidneys aren't functioning, they are becoming incredibly toxic. They may not even, their nervous system is shutting down on you, which means that all the rest are about to go, right? You've got to get those specimens collect those specimens in a quick hurry, spine or whatever you're collecting, urine, spinal fluid, I don't care what. You got to get the specimens quickly before you start your broad spectrum. Why? Because if you start your broad spectrum drugs, that before you collect the specimens, then you may never get your answer. You get what I mean? If you didn't know what you were ended up dealing with, you might not know. So you've got to be quick. And then start the broad spectrum, knowing there's no such thing as a safe drug, whether that's over the counter, whether that's prescribed over the counter or illicit. There is no such thing as a completely safe one. Broad spectrum ones are really not safe. They have a lot of things that can go wrong with them. Some of them can take your hearing out forever. Forever. So, but it's either like save this person's life or, whatever. So you do broad spectrum, and then you've got to get this identification quickly so you can back off of those big guns and give something that's more directed toward the thing. And usually that's going to mean it's less toxic for the patient. So if we hold on just a second to questions, we're going to see about this identification. So here's what we know in a micro lab. This is why it's so cool in a micro lab, because still, micro labs aren't being done by instrumentation. How many of you have been in a clinical lab? So some of you have been in clinical labs, and I don't know if they've been big labs or little local labs, but you know there's a lot of instrumentation there. I hear sometimes techs calling it machines. Machines run on gasoline, instruments run on electricity, okay? But anyway, so some tests, like in hematology and chemistry, it's really a lot of instrumentation. But in microbiology, it's not. It's that tech making sure that they know their organisms. Because they're going to have to be the ones that are discerning when they look at plated media or they're looking at stains, and we'll talk about that in a second. They have to know what might be a pathogen and what might not, and how to pull that out and get it so that they can get the information as quickly as possible. So, so anyway, just like not all colonies look the same of these potential pathogens, their sizes vary too. So when we think about the sizes varying, you can see some of this. Do I ever ask you specifics? I don't. If I haven't mentioned it, it would be something that you would need to know. But microscopy, I had wanted to go into lab today. I know some of you have a class after this, but I think I'll wait, if you don't mind, until we can start our Monday labs, okay? So, but we are going to go into lab, and we're going to see the different sizes of different bacterium. Some bacterium are distinctly different in their sizes and certainly different in their shapes and how they stain. So a microbiologist would be able to, truly within a couple of minutes of looking at a stain, would be able to tell whether there was probably a potential pathogen there or not. Just because they know, and whatever. So looking at those, but certainly bacteria are much tinier than fungi. And then fungi look very different than protists, right? So guys, it's important that we know the different kingdoms and whatever, because they all look different. And we're going to use the microscope as one of our first tools, especially on the original specimen. And I do want you to know that when I say an original specimen, it means a specimen that was taken straight from the patient. So that's what I mean when I say original specimen. It means the specimen that was taken from the patient. I just told you, I've explained to you why the integrity of that specimen needs to be maintained, right? How important that is. But what we also do, we don't just take that specimen and put it into media, enriched, differential, selective. We don't just do that. We will take a swab of that specimen and make what's called an original Gram stain. And I want you all to write that down, an original Gram stain. G-R-A-M, Gram stain. Original Gram stain. The Gram stain is the most important diagnostic stain in microbiology. It is the most important diagnostic stain in microbiology, the Gram stain. And certainly that stain that's done on the original specimen is really important because it can be done within minutes of receiving the specimen in the lab, and those results can get straight to that bedside. Now, so let me tell you what that means, all right? Original meant that it's from the patient, right? And we said that a Gram stain. I already talked to you about microscopes a little bit. I didn't really talk about resolution too much, but I will in a second. But what I'd like to get to now, and I'll come back to this, what I'd like to get to now is this idea about these stains. And what I want you to know about the stains is the Gram stain is the most important diagnostic one. It's a differential stain, which means it's going to be able to tell you the difference between different microorganisms. They're going to look different to you. It's not like everything that you put on that stain is going to be the same color, right? A simple stain is just going to be using typically one dye. Just one dye, which has its purpose, but a Gram stain is a differential stain, and it's going to have two different colors so that you can differentiate, hopefully, and now you're getting closer to figuring out what might be there as a pathogen. let me tell you about the original specimen gram stain. On that original specimen, we don't just see microbes. That's not the only thing we're looking for on an original gram stain. On an original gram stain, we're looking for white blood cells. Why would we be looking for white blood cells? Because they would indicate that there is definitely infection. Hey, look, we might not know yet what's going on, but guess what does know? Your body knows. Your body knows. And is your body trying to fight it? And one of the ways it fights it is by sending what to the site? White blood cells. So we look for white blood cells, and then we quantify them on the original gram stain. What does that mean? We give you an amount. So when you're reading a microbe lab report, it will say from an original gram stain, it will quantify how many white cells are there. It will also often quantify how many squamous cells are there. What would that particularly, what would that mean? Yeah, like skin cells or cells that were from, yeah, just the area, right? Suppose you had a ton of squamous cells and very few white cells. That would maybe indicate not a very good what? Specimen, exactly. And if you read that and interpret that, you might want to send another specimen, because that gram stain should have been sent to you in not too long, right? So you have a sputum culture and it's got a ton of squamous cells in it, very few white cells. It might be that you didn't really get sputum, you got spit. Does that make sense? So again, we're looking at the human cells as well. Suppose on the original gram stain, you see that there are a significant number of white cells, at least as many as squamous cells, and you see that there's some intracellular bacteria that have been noted. What does that mean? Intracellular bacterium in the white cells. It means that the white cells were done what? Well, they've eaten it. They've eaten the bacteria. So if you see on that original gram stain that there were intracellular microbes noted, and they'll give the gram reaction, I'll talk about in a second, you can bet that's a problem. I can tell you right now, I have never seen a case, well, I've seen tons of, I can't even tell you, hundreds of cases of gonorrhea and slides that were done with the original gram stains. And all of them had intracellular gram-negative diplococci on the original gram stain. They not right over them, but I'm just telling you, they do, because the person's body knew that that was a pathogen. So the white cells were doing what to those microbes? Eating them. If the white cells have eaten the bacteria, you want that bacteria. You want the ID on it. Okay? That's what I'm saying. And you can get that from an original gram stain. And how long does it take you after you sent the specimen? It takes minutes. It doesn't take long. It doesn't. Or it shouldn't take long. As soon as when the specimen gets there. Not all original specimens get a gram stain. Why wouldn't you want to do a gram stain, an original gram stain on a stool culture? There's so much stuff. There's not going to be anything that you're going to, really, there's just so much stuff. It's not going to help you. But on sputum, on wound cultures, these are important original gram stains that have incredibly important diagnostic purposes. Okay. Okay. Great. Now, so that's a little bit about stains, but may I, if you don't mind, I'm going to go back quickly to the microscope thing. We saw microscope. You guys labeled the light compound microscope. It's the most used microscope in microscopy in infectious disease, this scope. But it is not the only scope, okay, that's used in a real micro lab. So we have different types of microscopes that we are going to also use. I'm not going to care about too much resolution. I think y'all would understand if you want to be able to see something, right? So being able to use the microscope to be able to see it. But there are a couple of microscopes I want you to know about, and I want you to know why they're used in what I call a real micro lab, right? And one of those scopes is called a dark field microscope. It's called dark field. So you all were maybe seeing the other day that you could see the light coming up through your specimens, right? And your specimens were kind of stained. But in this, it's going to be kind of a reverse. So it's going to be this dark field microscope. And then I also want you to have heard of, and I don't know where it would be, I want you to have heard of a fluorescence microscopy. Fluorescence. Let me tell you where they're so important in a real micro lab. Is because sometimes we get specimens like spinal fluids. And a spinal, everybody understands that when a lumbar puncture, what people call a spinal tap, a lumbar puncture is done, should there be any microorganisms in spinal fluid? No, never. And if there's even one, is that a bad thing?
Yes.
Right, so it is. And so you've gotten a tap done where spinal fluid is leaking out. The last tube that they do, because they let some of them go because they know there's probably going to be a little bit of blood from the trauma of the tap. So the first one is, they get sent to different labs, but the last one gets sent to the micro lab. And in that micro lab, there will be an original gram stain done on that. And other things are going to be done. Why? Because do you expect to see a ton of things?
No.
Remember that urethral vaginal swab that had all kinds of mucus and white cells and bacteria? Do you expect that from the spinal fluid? No, there may be very few numbers of things there, but is it any one important to see? So if you did a stain and you looked at that, you could look for a long time trying to cover the whole field, and you would probably miss the few that would be present. But if you took a drop of that fluid and you put it, a cover slip on it, and you went into a room that is the darkest black you've ever seen, thought about. Like, it's not like night. It's like coffin, I would imagine. You know what I'm saying? So you can't even, it's these rooms, this dark field, for these dark field scopes, there's a little room with this scope in it, and you set your stage with your slide and stuff, and you get real ready. Like, you're actually already, you're sitting down ready to look, because you can't stand up and then sit down. It's that dark, right? You wouldn't know what you're doing. Like, you're already touching the scope and just hit the button. Goes completely black. So now if there's even a little bit of light being reflected to you, will you have a better chance of seeing it? Yes, you'll have a better chance of seeing it, catching it. So I do want you to know that dark field microscopy and fluorescence microscopy are used oftentimes in helping to get numbers when the microbes are in such low number from the original specimens that you want to try to get this. And they're also sometimes used for diagnostic testing as well, because we can specifically tag them. We don't have to write that. Just write down that in very low numbers. So we use other scopes, don't we? And then there's even an electron micrograph. If y'all know electron scopes, these transmission electron micrographs and scanning, we can see 100 times more than we see with the light compound microscope. But we don't use these in clinical microbiology. We do not use them in clinical microbiology, but they're used to help us to understand what is actually going on inside of cells. And how we know so much about what we do know about what's going on inside of cells is because of electron microscopy, right? Okay. So that's a little bit more about microscopes and a couple of different types of microscopes. We definitely sometimes want to use, instead of just stains, we want to use, and y'all are going to do this, I think, next week. So maybe this will come Monday, which means I would have to, okay, whatever. I kind of look at you. You guys are going to use wet preps. Do wet preps where I have living protozoa for you to look at, and you have to identify them. So I think it's going to be soon, as soon as I get them in. But you'll be looking at living protozoa using keys to try to help to identify them. Now, protozoa, proteins, they're huge, aren't they, compared to little tiny bacteria. So I think you'll have fun with that. But there are some things that we actually want to do wet preps even on original specimens, like vaginal swabs. Why would we want to do a wet prep on a vaginal swab? Yeah, but there are a lot of wet environments. But why with vaginal secretions, do you think? Does anybody think of something that we could have the identification just right there from the wet prep? No.
by the way it moves?
We use movement, but it's actually done for yeast. We can tell if there's like a lot of yeast. Have y'all heard of yeast vaginosis, right? So yeast causing problems. So you've got your ID right there. The person's probably still in the stirrups. You know what I mean? And you've got the swab. You're just looking and there's yeast. Or a protist. Does anybody know what protist we can identify from a wet prep within minutes of getting the specimen? You want to write this down? It's called Trichomonas vaginalis. It's a protist. So it's protozoa, isn't it? It's the most common sexually transmitted protozoa. People refer to this as a trick infection. Have you heard of that? It's called a trick infection. And it's called that because the genus is Trichomonas. The species is vaginalis. These are, compared to bacteria, these are huge. They are eukaryotic. They're huge and they swim. And they're very mobile. They're very mobile. They have flagellum. And they're really kind of cool to see and watch them swim. You know, so hello, there's your diagnosis, and there you go. Get flagell, and you're going to be good. You're going to be fine. What'd you say? So what's the question? So wet preps, like vaginal secretions, for wet preps are going to be great to do. Sometimes skin scrapings are going to be good to do with wet preps. And by the way, you all get to do this. These don't have to go to the lab. These are so common and so easy to diagnose that a lot of times you guys get to do this right there in the clinic or right there in the ER or right there wherever you're going to be looking. And you're not going to be afraid to use a microscope, are you? You're going to know how to use a microscope and you're going to be able to see these. So we can definitely do wet preps for those types of things. And then we certainly can do what are called stains. So did I add a couple things to your list today? What have I added to your list of microorganisms? Trichomonas vaginalis, which is going to be in that kingdom, the protist, right? And then what? I can't hear what. Haemophilus influenzae and Neisseria gonorrhea. So those are bacteria, aren't they? So we put those on our list. We start on our list. Remember, these quizzes can have anything from the list, right? Right. So stains. So stains can be done and sometimes on original specimens. And we said you should be paying attention to the results that come back from those original specimens. People should be paying attention. I wonder sometimes if they are. Sometimes I really do wonder that. But it's so important, and the information is so important with those. So I told you that some stains are called simple stains because they just have one dye, which means everything's going to be colored that same color. Your white cells will be blue if you use methylene blue. Your squamous cells will be blue, and all the bacteria present will be blue, right? Because the dye is blue, right? So anyway, that's just one stain. But if you've got a couple of different dyes and you treat it a certain way, which a gram stain does, it can be differential, which means that you're already getting toward your diagnosis. And that can happen from the original gram stain. I'm going to give you an example of how that actually happens. All right. So those are terrible examples. Okay. All right. So I really am going to give you examples, right? Because what can happen is this. Let's look at this little first picture. Can you see how some of these stains look purple? Or maybe they look black to you from there, but they're really purple. And some of them look pink. Do you see that? That's very easy to see. Some are pink, some are purple, right? So I do want you to know that if they're purple on a gram stain, they're called positive. They're gram-positive microbes. If they are pink, they are called gram-negative microbes. Fair enough? All right. So I want you to look at something else, though. And this is, I'm putting the root cart a little bit before the horse, but not really. What else? But if you look at these little ones that look purple, can you tell me anything else about them? Can y'all see them from the back?
They're round circular.
They look really circular, like spheres. They look like spheres, don't they? They do. And what about the little pink ones?
They're like elongated, like little rods. Do you think that gives us any idea about what they might be? And these ones that look purple, if you notice, they're in like little clusters, kind of. They're this one, not really, but most of them are in little clusters like grapes. They're not little purple spheres that are in long chains, are they? No. So do you think how they arrange themselves, the color and their shape play a role in us knowing what they are? Absolutely. As much as Jenna and Amanda look different from each other, they do too, right? They look different, and that means that we're already getting toward an identification, so we're happy about that. And so saying whether something's gram-positive, like, and I'll give you some examples of gram-positives, I won't ask you, but it's staph, strep, Clostridium, Bacillus, those are gram-positives. Gram-negatives is E. coli. I'm trying to think of other gram-negatives I'm giving you. Neisseria gonorrhea is gram-negative. So that is already differentiating my diagnosis by knowing what's there, right? Okay, so great. And then we have some other types of stains, and I do want to tell you about these other types of stains. Acid-fast stains are going to be absolutely necessary for a particular genus that we most worry about, which is Mycobacterium. And it's a Mycobacterium genus, tuberculosis species, that causes what disease? Tuberculosis. We know that tuberculosis species of Mycobacterium isn't the only species that cause tuberculosis. What else can? Did I have this on your list?
I don't think so.
Avium intracellulare. Mycobacterium avium intracellulare is also another species that could cause tuberculosis. But we certainly need to know about this, right? So guess what would happen if we just did a gram stain on that original specimen of a sputum? Would we be able to see this? No. So this is the reason, if we get a sputum culture, we need to know whether you're thinking TB. Because if you're thinking TB, we're going to add this acid-fast stain to the setup. Good?
Yes.
Okay, so great. All right, so we're going to add that to the setup, and it's going to be important that we do because that's going to be needed for that. Then we have some stains that we can do called endospore stains. So these endospore stains are going to be able to pick up spores, and not that many organisms make spores. So again, if you've got a positive endospore stain, are you narrowing down your diagnosis? Yes. Jenna, am I boring you? She's like, yes, move on.
I'm sick. I'm sorry. I'm like, yeah.
That's okay. I do want you to know that there are a couple that we really know produce these endospores. They are going to be the Clostridium genus. Clostridium is the genus, and Bacillus. Clostridium are anaerobes. And I do want you to know that Clostridium, all the species we hear about are anaerobes. What does that mean? That means that oxygen kills them, doesn't it? They can't grow in the presence of oxygen very well. So Clostridium, can you all tell me these species of Clostridium that we already have in our list? Did I give you all any? My new people, because I've given y'all some in A&P. All right, so let me give you a couple of species of Clostridium. You ready? Clostridium tetani. What do you think it causes? Clostridium botulinum. Botulism. It's a type of food. It can cause food poisoning. It can be from other things as well. And it causes a flaccid paralysis. It's got a neurotoxin. We'll talk more about what it's doing.
He says
Botulinum, Clostridium tetani, Clostridium botulinum, and Clostridium perfringens. My A&P people have had these. What is Clostridium perfringens? What does it cause? Gangrene. And y'all all know gangrene, right? With that. Okay, so we know that's gangrene. It also causes food poisoning. It's probably one of the most common, we've all had Clostridium perfringens food poisoning. We've all had it. I can guarantee that. That's how common it is.
fecal oral route transmission. If you've ever had something that you were violently, got sick all of a sudden, like you were well one minute, but sort of heaving the next, and you were really sick for a few hours, and then it all kind of went away, you were okay. It was probably that. Or staph aureus. So, guys, one more. We might as well add this Clostridium now. Clostridium difficile. Clostridium difficile. These are all anaerobes. These are in that genus Clostridium. Clostridium can produce endospores. And Clostridium difficile, what did y'all say? It's called C. diff, a C. diff infection. But what did y'all say that really causes? What does it cause? We know what tetani causes, tetanus. Botulinum causes botulism. Perfringens, gas gangrene, and food poisoning. And what did you say difficile causes? Well, it doesn't do it. The flora in your colon have been wiped out, usually because of antibiotics or some reason that's been disturbed. It's been wiped out by antibiotic use. And now the Clostridium difficile there's too much of it there, and the toxins it produces. It produces something called, it's called two different things, but I want you to know them both because you'll hear them both. It's referred to as causing antibiotic-associated enterocolitis. That's one word, enterocolitis. Antibiotic-associated enterocolitis. Now, why do you think it's called antibiotic-associated? Because the patient has a history of being on what? Antibiotics. And what does enterocolitis mean? It means inflammation of that colon or the enteric system, which could be even higher, right? And it does. It also causes something that we call, you ready? In ID, we call it pseudomembranous. One word, pseudomembranous enterocolitis. Again, that word enterocolitis. But what does pseudomembranous mean? Not a true membrane, so sort of a false membrane. The reason that we kind of call it this, we call this condition this, is because some of you all have worked with patients with C. diff, haven't you? They have terrible cramping, bloating, and really kind of uncontrolled diarrhea, don't they? And the odor of it should be diagnostic, but of course it isn't. It has to go to the lab. But wait a minute, listen up to this, because you do need to know. When you get a stool specimen on those patients, it looks like water with a green film. It almost looks like the lining of their intestines is coming out. Has anybody ever cleaned chitlins?
No.
Okay, if you've never cleaned chitlins, you would know that if you look, you turn them inside out like a sock, and then you really just, before you do that, you put a hose at one end, so all the, most of the big stuff goes out. But then you turn them kind of inside out, and you clean them out, and it's got like this membrane. And you have membranes on the lining of your intestines, but this causes almost a false membrane, and it looks kind of green. It's really, it's so awful. It's a horrible thing for those patients. It's horrible. Yes?
Don't people do fecal transplants for them?
They do. Well, that can be recommended sometimes. And this country is really regulated, you know. We have to, so we have to be careful about how, because let me say this, and I'll end it on this note. As far as infectious diseases go, who are you the most at risk from contracting an infectious disease? Is it your pet? Is it your whatever? No, it's from another human, isn't it? It's from human tissues, human fluids, human whatever. And so that's why it's so regulated, right? To think about. Do you have any questions for me? Do you have enough notes for a study guide? I think we really need, I wanted to show y'all these stains today, but what we're going to do in lab Monday, plan to stay for a long lab Monday, because I do have a couple of people who have Wednesday, this class and here Wednesday, and I don't want them to miss this. So Monday, plan to stay for a significant time, because we're going to be looking at these endospores, we'll be looking at Klebsiella stains I didn't even talk about. We're going to be looking at some of this stuff, okay? And if we get our protozoa, our living protozoans, you'll also be looking at those.
So no,
So no, I'm gonna wait so that everybody can be there. Be here Monday, don't miss.
Yeah. Can you hold just, let me turn this off. You can come right here.